Hello and welcome back to Simply Biology. Today we're going to be talking about amino acids. Now amino acids are possibly one of the most important molecules we have in our body because they are the monomers that make up protein molecules. Now let's get started. So as I've just said, amino acids are the monomers which make up a polymer or a protein. Proteins have a variety of functions including acting as enzymes, hormones, antigens, hemoglobin and cell membrane proteins such as intrinsic and extrinsic proteins. Amino acids are the monomers, or the small units, or small repeating units that make up a long-stranded polymer molecule, which is known as a polypeptide or a protein. We have the basic structure of an amino acid is shown here. We have a central carbon bonded to an amino group, which is an NH2 group, and you have a carboxylic acid group, which is C double bonded O, OH group. We also have a hydrogen atom in this R group, which is a variable group. This group varies depending on the type of amino acid. Firstly, the amino group is able to accept a proton or H plus ion to become NH3 plus or ammonia, and therefore an amino acid can act as a base. We also have the acid group. It's able to lose this proton or this hydrogen and form a carboxylate ion or a COO minus ion. Like I've written here, this amino acid is an alpha amino acid because the amino group and the carboxylic acid group are bonded to the same central carbon. There's also a type of amino acids known as a beta amino acid, which has the amino in the carboxylic acid groups bonded to a different carbon, but for our purposes we only need to know about alpha amino acids. Now there's also something else important to mention, which is why I've starred this carbon. This carbon is known as a chiral center because the carbon is bonded to 4 different groups. So this is one group, NH2, this is another atom, this is hydrogen, and then this is another group, the carboxylic acid group. Then once your amino acid has a variable R group, this group will be different, and therefore this carbon is bonded to 4 different groups, so it's known as a chiral center, and therefore this amino acid can show optical isomerism. However, the only exception to this is glycine, because this has an R group which is a hydrogen, and obviously that would mean that there would be 2 hydrogens attached to the central carbon, and therefore a carbon would only be bonded to 3 different groups, so therefore would not be able to show optical isomerism and would not be a chiral center. We also have, like I've just said, the properties of an amino acid. As a base, it can accept the H plus ions onto the amino group of the amino acid and form a positively charged amino acid or positively charged ammonium ion. As the acid, it can donate protons from a carboxylic acid group, forming a negatively charged carboxylate ion. So here is your negatively charged carboxylate ion, and here is the positively charged ammonium ion, and obviously these amino acids are both glycine because of the R group is a hydrogen atom. We also have the process of joining 2 amino acids together, which forms a dipeptide, and it's through a condensation reaction with the elimination of a water molecule. To separate a dipeptide, water is added, which is known as a hydrolysis equation. So we have 1 amino acid and 2 amino acids joined together in here to form what's known as a dipeptide, and as you can see, the COON is clearly labelled as the peptide bond. C double bond O to N is the peptide bond, and this bond will break during a hydrolysis where water is added across this bond to separate these 2 molecules, and obviously to form one amino acid and the other amino acid. Finally, you can also separate amino acids using their solubility, because every amino acid has a different solubility depending on the R group. Some amino acids have a greater affinity for the mobile phase, which is known as the eluting solvent, and will travel further up the chromatography paper, while others have a greater affinity for the stationary phase and will remain close to the bottom of the chromatography paper. Now, this solubility is dependent upon the R group, because each of the R groups are different, and therefore this will affect the chemical properties of that amino acid. Well, thank you for watching this video, and I hope you found it informative. Thank you. Goodbye.